Alright guys, it's Miss Scott. Ignore that. Okay, there we go. This is Miss Scott with your first set of notes for the biotech topic. Today we're going to talk about karyotypes. The three things I want you to be able to do by the end of this, I want you to be able to tell me what a karyotype is, what is a karyotype used for, and be able to read and tell information from a karyotype. So let's start with a review of what chromosomes are. So a chromosome are these structures that we find in the nucleus of our cells. It's basically tightly coiled up DNA that's wrapped around those histone proteins that hold all of our genetic information. This particular diagram shows a replicated chromosome. So you have two sister chromatids that are identical to one another. So these have undergone replication, ready for cell division. In humans, we, under normal circumstances, have 23 pairs of chromosomes in our body cells, or somatic cells. This would be things like our hair, our blood cells, liver, heart, etc., etc. Now, these 23 are not necessarily all equal. The first 22 pairs are called autosomes, while the last pair, the 23rd pair, are sex chromosomes, and these are either going to be X or Y. Again, just to remind you, the reason we have pairs is because we get one set from mom and one set from dad. So altogether, we have 46 total chromosomes in those 23 pairs. Now, where do the karyotypes come in? A karyotype is basically a picture of an organism's chromosomes. And these chromosomes are paired up. They're paired with their homologous partner. So the one from mom and one from dad, and they're sorted based on their size and their banding patterns. The banding patterns that you'll see on the examples coming up, those show where it shows the location of genes. And on the homologous chromosomes, remember, you're going to have the same gene locations, but you might have different versions of that gene. In a karyotype, the autosomes are always paired up first, and the sex chromosomes will always be shown last. They'll be in the bottom right-hand corner. So here's a picture of a karyotype. Again, you see the autosomes here in red. They're sorted by size. The first chromosome is always the largest, and as they go on, they get smaller and smaller. Here in blue, we have our sex chromosomes. In this particular example, we have one X and one Y. Usually, in a karyotype, it'll have, in addition to the numbers, it might also have X and Y here, and it'll be easier to tell which you have. If you don't have that, though, a way to tell the difference between X and Y chromosomes, the X chromosome is always larger. The Y chromosome is significantly smaller than the X. So that's what it is. So now, why is it so special? Why do we have interest in it? Why do we need it? The benefit of having karyotypes is that we can use them as a diagnostic tool. We can use them to detect disorders that are caused by having the incorrect number of chromosomes. Now, why a person might have an incorrect number of chromosomes is because of non-disjunction. And remember, non-disjunction, non and dis both mean not, so this is not, and junction is together. Basically, this is where the chromosomes, during meiosis, they fail to separate, and you end up with either a sperm cell or an egg cell that has, instead of having 23 individual chromosomes, it has either too many, say 24, or too few, say 22. So non-disjunction causes these disorders that we use a karyotype to detect. We can also use a karyotype, in addition to looking for those particular disorders, it can be used to determine the sex of an individual. So in females, there will be 2 X chromosomes, and males will have an X and a Y. So here's a normal female karyotype. Some karyotypes, you may also note, they'll have a number and then 2 letters. This number represents the number of individual chromosomes. So again, normal for humans is 46. And then these 2 letters tell us the sex chromosomes. In this case, XX, and we see those down here. These are relatively the same size, this would indicate that this person is, biologically speaking, a female. And for comparison, here's a normal male. Again, if we counted all of these, there would be 46. There's 2 for each of these numbers and then 2 sex chromosomes. The only difference that we see here, here's the Y chromosome. Again, as I stated earlier, this is a lot smaller than the X chromosome. This is actually missing certain genes that is also carried on here. And we'll talk about that more when we get to genetics, talking about certain sex-linked traits. Fun stuff. So non-disjunction can happen on any chromosome. There's no reason it couldn't, but a lot of the times if a zygote, so remember that first cell after fertilization, if it does, for a lot of these chromosomes, if there's too many or not enough, generally that's what we call a fatal condition, so that embryo won't develop, or the fetus won't develop, which results in miscarriages. But there are a few disorders that individuals can have that we do see in the general population. So you can be born with these. The first one is Down syndrome, which you're probably familiar with. It is also called trisomy 21. Again, that tri refers to 3. And this particular one is caused by having an extra 21st chromosome. With Down syndrome, this can occur in both males and females. There's no one particular ethnicity or economic status or anywhere where it's more common or more rare. It occurs in 1 out of every 691 births in the U.S. And the incidence rates of Down syndrome do increase with maternal age. Basically, the older the mother is at the time of birth, the more likely the chances are that her child will have Down syndrome. So here's a karyotype and some examples of individuals who have Down syndrome. Again, we can look and see all the chromosomes sorted out. Here are those banding patterns I talked about. Again, they should match up on the homologous ones. And if you look down here at 21, there's 1, 2, 3, there's your trisomy. This particular karyotype, again, not only can we tell that this person has Down syndrome, but also their sex. In this case, this is a female because they have 2 X chromosomes. Okay. Our next disorder that we can use a karyotype to detect is called Turner syndrome. And in contrast to Down syndrome where you have too many chromosomes, Turner syndrome is caused by missing a chromosome. In this case, these individuals have only 1 X chromosome, so this is a sex chromosome disorder. Because you have only an X chromosome, this is only going to occur in females. It is much more rare than Down syndrome. It occurs in 1 out of every 2,500 female births. So again, we don't necessarily account for males in this because it only involves the X chromosome. Some of the symptoms that you may see in individuals who have Turner syndrome: a lack of ovaries and secondary sex characteristics. Often you'll see that there is extra skin on the neck. It's called a web neck, which we'll see in the next slide. Generally, these individuals are shorter in stature. They're prone to skeletal abnormalities in their legs and their arms, and they are more prone to having heart and kidney problems. So here's a picture of the karyotype as well as 2 individuals who have Turner syndrome. We can see, again here, if you counted all of these chromosomes, there's only 45 because we're missing this X here. There's only 1 X chromosome. And our last disorder that's caused by non-disjunction that we can use a karyotype to detect is called Klinefelter. This also involves the sex chromosomes, but instead of only having 1, like in Turner syndrome, this is caused by having 3. In this case, you have 2 X's and 1 Y. Because there's a Y chromosome involved, this will only occur in males. It's still counted as a pretty rare disorder. In 1 out of every either anywhere between 500 to 1,000 male births is going to affect. Some symptoms with Klinefelters that you may see, generally Klinefelter males tend to be taller, they have broader hips, they might suffer from learning disabilities and underdeveloped sex organs, and most commonly infertility. It's actually not terribly uncommon for people with Klinefelters to not know they have it until they're into adulthood and they realize that they are infertile and they go to a doctor and see why, and then they realize they have this extra X chromosome hanging out. So, here is our karyotype, what you're going to be looking for. Again, everybody is paired up. This is all normal, one from mom, one from dad, and then you get to the sex chromosomes and you have 3. Again, the X's are much larger than the Y chromosome. That's your giveaway. So let's test yourself. I'm going to go through, I have 5 karyotypes. For each one, I want you to see, can you identify the sex? Are they female or male? And are there any chromosomal abnormalities of the 3 we just mentioned? So I will show you the picture. I want you to pause it when you think you're good. Go on and I'll give you the answer. So our first one, this is a Klinefelter male. We can tell that because they have 3 sex chromosomes, 2 are X's, and 1 is a Y. And we know Klinefelters are always male. Our next one, this is a karyotype of a chromosomally normal female. 2 ways you can tell that. You could have just looked down here in the corner. 46 chromosomes, 2 X's. Everybody is paired up. There's 2 of each, no extra, none missing. Our third is a male who has Down syndrome. Again, what we're looking at here, it's 21st. There's 3. Male because I have 1 X chromosome and 1 Y. Our fourth, this is a Turner female, or a female who has Turner syndrome. Again, if we look, all the autosomes, these body chromosomes, are paired up, except the sex chromosomes where I only have 1. And our last one, this individual is a male who is normal chromosomally speaking. Again, I can count there are 46 chromosomes, and 2 of those are sex chromosomes. 1 is X, 1 is Y. If you have any questions, I want you to write those down in your notes and bring them to class tomorrow. If you're not in my class and you're using this for your own benefit and you have questions, you can leave them in the comments for me.